Dear students, I hope you're all well and I hope you read these experiments and understand well, which we discussed in last week. Today, we are going to discuss about the types of germination. The process of germination is categorized into two types depending upon the behavior of cotyledons during germination. First is apigeal germination and next is hypogeal germination. So in apigeal germination, as name api means above, in this type of germination, the cotyledons are lifted above the ground due to rapid elongation of hypocotyl. The cotyledon turn green and form the cotyledonary leaves. It is found in beans, castor, papaya, cotton, sunflower, tamarind, etc. Okay. The various stages during apical germination of bean seed are summarized as follow. First is under unfavorable conditions, the seed absorbs large quantities of water and swells up. The seed coat softens and ruptures at the radicular end. The radicle pierces out of the seed coat, bend downwards and grown into the soil. The radicals forms the primary root, lateral root begin to emerge, root hairs appear in the region where elongation has ceased. Water and salts absorbed by the roots hair are passed on the seedlings. Meanwhile, the hypocotyl elongates and arches upwards above the soil. The two cotyledons are within the ruptured seed coat. The hypocotyl strengthen, pulling the cotyledons into the air. The ruptured seed coats may remain below the soil or are set above the soil. The hypocotyl above completely erect. The two cotyledons diverge to form the flat green cotyledonary leaves. The epicotyl elongates, bearing its tip to the plumule. The young leaves unfold at the tip of the epicotyl. The cotyledons shrivel up and are set. The primary root gives branches and persists all throughout the life of the plant. Such a root is called a taproot. The young leaves now prepare food for the plant. So you can see that how critical processes is that the germination of a seed. Okay, so beginning of a new life is not a easy thing. You can see the images when you read the different stages. Okay, in first stage you can see it is swells up somehow. Okay, and radicals is just go downside. Okay, in this, but as it is the apicotyl germination, so you can see that broken seed coat is from here. Okay, and curved hypocotyl comes above the ground. Okay, so like this, one primary root tap root is formed here. Okay, and secondary roots also arise from it. Then, so you can see here that apicotyl is above the ground and remains persist throughout their life of plant. Note, in endospermic seeds like castor, the cotyledons enveloped by the endosperm and testa rise into the air. The cotyledons absorb all the food reserved from the endosperm, which is then fed along with the testa. The castor seed contain kernel, whitish structure at the tip of the seed has greater water absorbing capacity. So next is hypogeal germination. When the cotyledons remain under the soil or just on the ground during germination of seed, it is called hypogeal germination. It is because of the rapid growth of epicotyl that the plumule emerges out of the soil. It is found in the seeds of gram, pea, maize, mango, groundnut, etc. Various stages during the hypogeal germination of maize, a monocotyledonous endodermis seed, and P, a dicotyledonous non-endospermic seed, are summarized as follow. I hope you remember these terms. Okay, endospermic seed and non-endospermic seed. We already discussed in last chapter also. Hypogeal germination in maize. In the grain absorbs water and swells. The cotyledon absorbs food material stored in the endosperm. Then the radical passes through the protective coat, sheath, coleorhiza. It grows downward into the soil. The radicle develops into the primary root with root hairs. The coleoptile elongates and pierces out of the grain in the form of a tube opposite in the direction of the coleorhiza. So you can see the figure also as the stages mentioned in first, second, third, fourth, and last fifth. In fifth condition, a cluster of fibrous root arise around the base of the primary root. They develop from the base of the stem. The first leaf of the plumule passes out of the coleoptile, which now forms a tube-like collar around its base. Okay, you can see that a tube-like collar around its base also. More leaves appear in succession. Primary root persists, giving rise to fibrous root system. Okay, so you already know that in monocotyledonous plants there is a fibrous root of system is found. So there is no main root, only fibrous root or adventitious root is found. Then hypogeal or hypogeal, you can say germination in the seed of pea. The seed absorbs water and swells. Food reserves in the cotyledons become soluble by the action of enzymes and pass in the tips of the growing plumule and radicle. Then the apicotyl begins to elongate and forms a small loop. Root hairs appear in the region of the radicle where elongation has ceased. The apicotyl elongates rapidly. The plumule located young leaves appear and prepare food for the plant. Seed coats and cotyledons remain below the soil. Primary root persists, giving rise to a taproot system as it is a dicot plant. So, if you think about the difference between these apigeal and hypogeal germination in two lines, so first is cotyledons come above the soil, and in hypo, as name suggests, hypo means below. Cotyledons remains below the soil. Hypocotyl elongates and apicotyl elongates in hypogeal germination. Okay, these two differences in them. So another type of special type of germination is viviparry. The seeds of the plant growing along the seashore or in a saline marshes show a special type of germination called viviparry. In this type of germination, the seed germinates inside the fruit while it is still attached to the parent plant. A club shaped radicle emerges out of the fruit. The lower part of the radicle gets swollen. Finally, the seedling gets detached from the parent plant and gets embedded in the muddy soil below. The radicle develops lateral branches quickly to anchor the seedling. This type of germination is found in rhizophora, Syropsis, Avicennia, Heritaria, Soneratia, etc. Okay, so viviparry is one example that you can see that germination. First, you see the stages germination while in fruit. Okay, first stage, second attached seedling with fleshy radicle, and third detached seedling with lateral roots. If you see the figure also, okay, you can understand well. Mainly seeds of plants growing around seashore or in saline marshes show a special type of germination called viviparity. Okay, this type of germination mainly shown by these plants, Rhizophora and Heritaria. And there is some key points which maybe you don't know earlier. Okay, raphe as a prominent ridge-like structure that represent a part of the stalk. Okay, seed you already know that fertilized mature ovule, and tegmen inner thin membranous seed coat. Okay, and testa is outer hard protective seed coat is known as testa. So I hope you will be understand that these types of germination, and if you have any confusion you can ask to me anytime. Thank you.